Good morning, students. In previous lecture we have studied gene transfer methods, among which physical and chemical methods we have studied in detail. Today vector mediated and specifically Agrobacterium mediated gene transfer method will go in detail. So this Agrobacterium is a soil bacterium and this is gram negative. It infects the plant cell near the wounds, usually at crown of a root which is near to the soil surface. This is natural genetic engineering because it has ability to transfer the tDNA, which is part of a TI plasmid, and that is present naturally in this Agrobacterium cells, and it forms a crown gall. There are two main species, Agrobacterium tumefaciens, and this is with a TI plasmid, whereas Agrobacterium rhizogenes it is with a RI plasmid. And this plasmids can cause either tumor on the infected plant or hairy root disease on the infected plants. This TI plasmid, it is of length 200 kb and having T region which is of length 23 kb, that is transferred into the plant cells and integrated into their genome. The foreign gene which is of our interest, it is cloned in the tDNA region of this TI plasmid in place of unwanted sequences. Then we'll go in detail about the TI plasmid which is present in this Agrobacterium tumefaciens. This TI plasmid, it is with regions like tDNA which is useful to transfer into the plant cell. So we can incorporate in this region a foreign gene. Then virulence region, that is again essential for virulence, specifically for induction of a tumor or hairy root into the infected cell. Then third one, that is opine catabolism region. Then consecutive transfer, that is ORIT region, and obviously a character of a plasmid, that is origin of a replication. Only because of this it can replicate into the cells. Then this tDNA, again it is with the three genes of which TMS1 and 2, they are responsible for shooty locus, whereas this TMR is responsible for rooty locus, and that can synthesize the phytohormones like auxins and the cytokinins. And this locus or these genes encode the enzymes. Obviously these are responsible for synthesis of the phytohormones like auxins and cytokinin, and because of which there is a formation of a crown gall, or we can say tumor, into the infected cell. Along with this opine synthesizing region which is present in the tDNA. And what are opines? We must know. So these are the amino acid or sugar derivatives. Specifically these opines are synthesized by this Agrobacterium and not by the plant cells. These opines are derived from the arginine and the pyruvate that are found in the plant cell. Common opines are octopine and nopaline. Along with this, again this OS region that corresponds to the enzymes like octopine synthase and nopaline synthase, which are coded by this tDNA. Then opine catabolism region. So whenever TI plasmid encodes octopine, the plasmids are octopine type, and if they encodes for the nopaline, they are nopaline type of a TI plasmid. Interestingly, higher plants they are not able to utilize these opines, whereas Agrobacterium can effectively utilize these opines, and they are as a source of a carbon and nitrogen. So the tDNA region of all Ti and Ri plasmid, they are flanked by perfect 25 base pair direct repeat sequences. Means this tDNA is with right border and left border of the 25 base pairs, and that is essential for tDNA transfer. Then another interesting part of this Ti plasmid is vir region or virulence region. This is of 36 kilobase pair and composed of 8 operons like A, B, C, D, E, F, G and H, among which A, B, C, D are virulence or they are responsible for virulence, and C and E for the tumor formation. Of these 8 operons, Vir A is a chemoreceptor, and that senses the presence of phenolic compound. Whenever the plant is wounded, the phenolic compound is secreted by the plant tissue, and it can be or it is identified by this Vir A region. Then this Vir A region gives the signals to other genes like G and remaining genes, that is C, D, E, F. So it is activator. Vir A is a chemoreceptor or activator, and from which other genes are activated. But only these genes are not able to transfer. They need this 25 base pair direct left and right borders of the tDNA. So activation is important. This is additional figure that gives you idea about the tDNA. Again, already we have discussed, it is with the TMS1 2 region, which is responsible for synthesis of phytohormones. Then NOS region for the nopalin biosynthesis. This 25 base pair left and right border sequences, which is site of endonuclease action during the tDNA transfer, and the only sequence of tDNA essential for its transfer. Along with this Vir region, it is with Vir A, which is receptor for acetylserine. Afterwards, that activates other genes, and along with these 24 base pair right and left border sequences, that enters into the plant cells. Then this is additional diagram that gives you what is the exactly mechanism behind Agrobacterium with the plant cell. Here the plant, if it is wounded, obviously it produces acetylserine, and these acetylserine activates the Vir region or virulence gene, and these Vir proteins, obviously that Vir gene, which is already told that 8 operons are there, and these Vir genes after with Vir A region that synthesize single stranded tDNA, and this single stranded tDNA enters into the plant cell, obviously into the nucleus and chromosome of the plant cell. So this tDNA, it is with opine synthesizing region, then auxin synthesizing region, then cytokinin synthesizing region. So with the help of this tDNA, plants are going to be synthesize these phytohormones. Already plant, they are with the phytohormone, they are having the sites for the synthesis of the phytohormones. Along with this, the tDNA region, which is with these auxins, cytokinins and opine. So there is excess production of phytohormone, and obviously because of this there is a formation of a tumor in Agrobacterium infected plant cell. And this is a figure of the acetylserine gene that gives you idea about how this is activated with the help of the acetylserine gene. Here, this is a diagram in brief how this Agrobacterium tumefaciens bacteria which is with a TI plasmid, and whatever the foreign DNA which we want to incorporate into the plant, so we have to enter, or we have to cut first this TI plasmid at tDNA region with the same restriction enzyme DNA of interest is cut, and then recombinant TI plasmid is obtained. Then this recombinant TI plasmid is transformed with the Agrobacterium into the plant cell. How it is transferred, it is discussed in the previous slide. Then transformed plant, it is now with TI plasmid, and the tDNA carrying region of interest is integrated now into the plant chromosomal DNA. Now this is a cell that is with inserted tDNA carrying gene of interest. Now these cells are cultured on the selective medium, and a transgenic plant is obtained from a specifically transformed cell. Now this infected plant shows the genes of interest which can be expressed as a new trait, and it is identified with the help of this tumor or hairy root disease if it is a Agrobacterium rhizogenes, and there is a tumor formation if it is a Agrobacterium tumefaciens. So with this, how this Agrobacterium mediated gene transfer is obtained is over. Thank you.